Thank you. So we have 2 short talks in the program. The first one is by Florian Ströhl. He comes from the Arctic University of Norway. His talk is titled Instant Volume Microscopy, and he will present solutions to reject the out-of-focus light. Welcome.
Thank you very much. Hi. So thank you for giving me the opportunity to talk here. As you said, I'm from the Arctic University of Norway in Tromsø. This is the island where we are. And what we like to do there is, especially in winter, fluorescence imaging. And one person that is very interested in doing that in collaboration with me is this person, Åse Birgisdottir, and she's very interested in the heart. She works in a hospital. And she works on human heart tissue, and she has a wonderful system where she takes induced pluripotent stem cells to make these types of tissues. They're beating, they're 3D, they're alive. The scale is 1 millimeter. It's huge as far as we are concerned. And unfortunately, she is also very interested in mitochondria, which are on a completely different scale. So imaging these is a challenge. And I'm in optics, so we work together and try to find a solution for that, getting some 3D fast imaging of mitochondria in really hard-to-image tissues that are not cleared. So for that, we look at 2 different techniques, multifocus microscopy, to get whole volumes imaged in a single shot on a camera. But because it's unclear tissue, we also need some optical sectioning. And we combined this together into the third part of the talk, which is about the instant volume microscope then. So very briefly, multifocus microscopy is when you take a regular microscope like this, and you just replace the optical parts in front of the camera with multifocus optics. That's it. There have been a ton of publications on that. Light field microscopy is one where you use microlens arrays to get a light field. You can use beam splitter cascades that multiplex your image. These nice prisms are also something you can find in literature. And also gratings that split your light into different focal planes. That's the darling of Sarah Abrahamson, who might be a name to some. And they are all quite nice techniques. They illuminate, they record every plane that is illuminated. It's nice. They're very fast. The sample is stable, so you don't have to move it around. If it's a watery sample, it might just slush around. And really important is you can use conventional sample mounting. I'll come to that later. And they're also not too expensive. On the downside, of course, you have this out-of-focus haze. The field of view gets smaller because you put all of these planes on your camera at the same time. And especially for light field microscopy, you need reconstruction. Let's cut out light field microscopy. And with bigger cameras, we also don't need to worry too much about a small field of view. But this out-of-focus haze, that is still a problem of all multifocus techniques. So how do we get around that? Well, traditionally, if you want a section image, you have a sample, different planes that illuminate light. Take a knife, you cut it. That's called sectioning. It's deadly. So that's not useful for us. You have other options to use. The most prominent one is probably confocal microscopy. You take a single point of illumination, and then you have a focal conjugated spot that blocks all the light. And it works, but it's quite slow. And the gold standard for this type of imaging is usually light field microscopy, where you come from the side and you image orthogonally to that. The problem there is you have 2 objectives, and that can be troublesome if you have a sample that is bigger than the volume you want to put this into. And if you want to look at mitochondria, you need really high NAs, but at the same time, we have a really big sample. So we have trouble fitting it in there. So we decided to take the idea of light field microscopy and combine it with multifocus microscopy. And what we came up with is the following. So this is a regular multifocus microscope. And now we just put a Galvo mirrors there and change our illumination to be a light sheet. Just comes straight out to the objective. It's not orthogonal. And we just scan that. And let's see what happens. So as you scan it through and the camera is just exposing during that time, you end up with basically wide field illumination. So you have all the autofocus lights still there. So that does not work. What you have to do is something else. And you can read about it in a bit more detail in the archive paper that got out, I think, 2 days ago. And that's, we put a bit of drizzle of confocal microscopy in there. Think of it like this. If you connect your light sheet with your camera, and you would move the camera along, that means the PSF of the camera moves along with the PSF of the light sheet. Then in the math of the formula, these two move together, and under the convolution, they multiply. So you get the light sheet stacking effect also with a light sheet that propagates along the direction of the light sheet. In simpler terms, we have a rolling shutter that is a pinhole. And because we are imaging all the different planes at the same time linearized onto our detector, this rolling shutter is everywhere where the plane of illumination is. And as we scan across, the rolling shutter pinholes all of our different plane positions. And when the camera is done exposing, everything is optically sectioned. If some people here know structural image microscopy, you might know that that is a trick you can also use to get additional resolution. And that's exactly what we did by just angling our light sheet a tiny bit. Here's the effect of combining the light sheet along the optic axis. When you tilt it, you get the same effect, but it stretches it along the axial direction. So you actually increase your resolution also along the axial direction with that. And it's optically sectioned. When we do simulations on this technique, this is regular wide field microscopy. This is cut through the optical transfer function. And by using this technique of using a tilted light sheet and synchronized readout with the camera, then we actually get a much higher resolution along set and basically a rejection of all the autofocus light. It works in real life as well. So these are beads. And you can see that this was a beam splitter cascade multifocus microscope. And we have 3 different focal planes. And they all depict the same bead in different focal positions at the same time. In real cells, it looks like the following. So this was done with a 40x objective that makes the magnification changes how far these planes are apart that we're imaging. So these were plus and minus 12 microns apart for the nominal focal plane. And you can still see there's a lot of out-of-focus light in these planes. These are very thin PPAE cells. And if we turn on the synchronization between our illumination and our detection, then all of that background light disappears. And even when you look closer, you can see that the image actually becomes a bit crisper as well in the focus plane. And that's this frequency mixing effect that you know from structural image microscopy. We can do that quite fast. So this is by moving the stage at 35 volumes per second. And in principle, so these are 3 planes, but nothing stops you from taking a 3x3 and 9x9 plane image. And you basically build up more and more focal planes as you wish, as long as your camera supports it and as long as you have enough signal. So with this, we felt brave enough to actually try it out on these hard tissues. And so this is a zoom through where we go from the get-go at the cover slip, and then we go further and further into these tissues. And you can really see up to about 25 micron in this unclear tissue, the mitochondria. That's on your right side. On the left side is the equivalent view with a regular multifocus microscope, where it is actually quite challenging to see mitochondria if you're only a few micron deep. Every of these planes that I just showed you in the zoom through is actually a 2 micron thick volume that displays the mitochondria. And if we have a color-coded view, then you can really see in a single cell, it's one shot, you can assign all the mitochondria in a cell to exactly the set position where they are within the resolution limit of the system. which is about 500 micrometers. And that works actually quite deep into the tissue. So on the right side, there's a small inlay which shows a round little thing. That's a mitochondria-derived vesicle. And that has a resolution of about, or depicted resolution of about 300 by 300 by 500 nanometers. And that's pretty much resolution of the system. What is important, though, is that we can see it in the first place. I needed to actually use the image on the right to find it in the wide field image because it just wouldn't appear otherwise. It was quite difficult to spot. Okay, and with that, I want to wrap up. So I told you about multifocus microscopy, optical sectioning, and how we combine the two to have instant volume microscopy. And I, of course, didn't do that all on my own, but I had a big, helpful team on the side. There's Daniel and Jon Rickard, and also, of course, Åsa and Mireia with the hard tissues, and also the rest of the nanoscopy team at UIT, Diana, Oliver, Marie, Ida. And I also would like to thank the funding buddies, especially the Norwegian Forskningsrådet, who funded the project, and I also would like you for your attention.
Thank you. That was wonderful. Do we have questions about this technique in the audience? Here we have one.
Well, I don't know if it's compatible or what. I'm not an optician, in optics, sorry. But I was wondering, would you have an advantage in using a Bessel beam or an Airy beam?
It's a good question. Potentially. The way you make this light sheet is you illuminate, when it's angled, you illuminate only a part of the aperture. And what we are currently using is a truncated beam that illuminates pretty much half the aperture. That's exactly the volume that we're imaging. So by using a different beam, like a Bessel beam, you would usually make the beam longer, but we are kind of limited anyway what we can use. So potentially have a benefit. We haven't tried it out, but my guess would be that it has not too much of implication in this case. Maybe if you want to go to much thicker tissues, it becomes a lot more influential.
Okay, thank you. More questions? One there.
Yeah, great talk. So the question is, the whole structure is beating, so you have to synchronize it because still your imaging speed might not be high enough to get rid of any artifacts because the whole structure is moving. How do you do that?
So you could synchronize it, but the 35 volumes per second, that is still not the maximum. That's just what we did. You could go faster. That's also possible. It just depends how fast your galvo can go, for example. If you would want to, if you have like a heart that's super fast beating, this is actually, I think, 1 hertz beating, so that's not too fast actually. If you have something that beats much faster, which is perfectly possible, then you probably need to have some kind of readout that tells you that. I mean, you can try to use image-based, so you have a run and you see it's repetitive with a certain frequency, then you might be able to lock it in. Otherwise, you probably need some kind of ECG or something that reads it out at the same time. For heart. If you have a different system, of course, it might be different.
Thanks.
Okay. Thank you very much once more.